Good morning and Assalamualaikum everyone. So we proceed to lecture 12 on microbiological test. We have learned about the microbiological criteria. So the microbiological criteria, they tell us the limit for microbial counts. Okay, so how do we know, or how do the industry make sure that they follow the microbiological criteria? So they need to conduct microbiological tests and analyze their results. Okay, so microbiological tests are used to detect and monitor the presence of harmful pathogens in food products and in the processing environment. So please take notes that microbiological tests are not only used to detect and monitor the presence of pathogen in food product only, but also in the processing environments. Maksudnya, you also can conduct microbiological tests for the surface, either the surface is clean or not, on the operator's hand, either their hands are clean or not. So you can do some microbiological tests, okay, to see, to detect the presence of pathogens. So they are classified into three major types, which is the culture dependent assay or the culture method. Okay, the second one is the rapid immunological or antibody based assay, and the third one is the nucleic acid based assay. So for the method number 2 and method number 3, these are not covered in your syllabus, so this is also not relevant for your examination. Only we will cover only the culture method, okay, the culture dependent assay. So the culture-based assay is also known as a standard plate count technique or standard cultural techniques, or sometimes we just say the culture method. Okay, the principle is that when you have a material that is contaminated with the bacteria and you culture them in the lab, okay, the viable bacterium develops into a visible colony on the nutrient agar medium. So we culture them on the nutrient agar medium, as you can see in the YouTube figure here. So it is a well-established method and mostly used in microbiological lab. So the advantage is that it is least expensive, it is easy, and you are able to isolate the live cells of the pathogen that you want. Okay, for example, you want to test for the salmonella, you can use the culture-based assay for the salmonella method. Okay, so the disadvantage, it is very time consuming, so it takes time for you to get the results. Okay, so there is a further reading, there is a link for the further reading on this culture-based assay, and also a video in Putra Blast related to this. How to do this, the procedure on how to do the culture method is not relevant for your exam, but it is important for you to know, because later on when you are working in the food industry, you will always come across the microbiological test. Let's see how the industry conduct a microbiological test to monitor the microorganism in their food factory or in their food products. Okay, so we can differentiate between two procedures for the microbiological testing in food industry. The first one is the food testing, meaning you want to test the food product or the raw materials, either is it whether it is safe or not. And the second one, you want to test whether the factory surrounding, okay, the factory surfaces are clean or not. Okay, so there are different microbiological contamination that can happen in food plant, and that's why you want to monitor through the microbiological test. The first category is the indicator organism. The second one is the spoilage organism. The third one is the pathogenic bacteria, which are harmful to us, for example, Salmonella, E. coli. And the fourth one is virus. The fifth one is toxins. Okay, so our concern is actually on the pathogenic microorganism. These are either not allowed at all in the foodstuff, or they are limited to a specified number of cells per gram of food, meaning they have a limit, okay, in their population. So these limits are different for different food products. Nevertheless, the most important thing that we have to remember, if the limits are exceeded, this means the production of the food can bring adverse consequence for the consumer's health. Okay, usually we have a zero tolerance per 25 gram of food sample for the pathogens as our limit. Okay, maknanya selalunya limit yang diberikan kepada patogen ialah zero, absence in 25 gram of food sample. Okay, tak boleh ada pathogen dalam 25 gram of food sample. So as you can see, figure here is the microbiologists, okay, taking samples of food, minced meat, okay, daging hancur, and then they want to conduct the culture method, okay, the standard plate count test for the microbe, to detect the microbe. Okay, here is an example of microbiological test that is conducted for the food. Okay, so the food here is the juice. Okay, so we want to conduct E. coli test for the juice because the presence of E. coli 0157 is usually monitored for the pasteurized concentrated orange juice. Okay, scientifically it has been established, or it has been stated that juice must be heated, okay, or must be pasteurized to 72 degree for 3 seconds so that E. coli can be effectively eliminated. Okay, this is a scientific statement that you want to comply in your practice for the pasteurization. Now, the effectiveness of the heating process in eliminating the E. coli must be validated. Okay, so validation is the way how you prove that the pasteurization has been correctly conducted and able to control the hazard. Okay, here hazard is the E. coli lah. Okay, hence the juice sample will be taken from the processing line. Okay, will be sampled from the processing line and then sent to the lab for E. coli analysis. Okay, the result, it is very important for the lab to provide evidence that the heating or the pasteurization has eliminated the E. coli and comply the microbial limit. So the microbial limit for E. coli in juice is usually absent in 100 ml. Maksudnya untuk kita membuktikan bahawa proses pasteurization kita telah berjaya menyingkirkan E. coli, kita kena tunjukkan melalui microbiological test that dalam jus kita tak ada E. coli in 100 ml sample. If this is proven through the microbiological analysis results, then your pasteurization is considered effective and your product is declared safe. If the limit is exceeded, the process is not effective and the juice will not be acceptable, so the whole production will be stopped and investigation will be carried out. Okay. The second microbiological test that is usually conducted in food industry is the hygiene monitoring, okay, or the environmental monitoring. This is a microbiological test that is conducted in the surrounding of food production. Okay, bukannya dalam makanan itu sendiri, tetapi keadaan dalam facilities sekeliling production. Okay, why? Because there has been a number of foodborne illness outbreak that happen because of poor hygiene practice. Okay, mungkin disebabkan oleh lantai yang kotor, okay, ataupun tangan operator yang kotor. So microorganism from these surfaces can cross-contaminate the food products and lead to foodborne illness for the consumers. So microorganisms, they can survive in the environments. Okay, not only in the food product, they can survive in their environments, and usually they are brought into the food environment or into the food factories through raw materials, okay, pest, air, water, and also employees. Okay, but however, we are able to control this microorganism if we apply a good sanitation practice, okay, good cleaning program, and there have been quite a well-established histories, okay, where pathogens, they are able to survive in the environments of food factories and then brought by air or by operators from one surfaces to food products. Okay, for example, listeria, they can grow in the floor drain, okay, sistem saliran dalam kilang makanan, and then they can be brought to the food products through air. Okay, this is an example of environmental monitoring program. So environmental monitoring program is a method or it is a practice to assess the effectiveness of the hygienic practice in a food factory. Okay, how to implement EMP? EMP is usually applied to several zones in the food factory. Okay, for example, here you have zone 1, zone 2, and zone 3 and zone 4. So zone 1 is the blenders, conveyors, utensils and work tables. Okay, zone 2 is the equipment framework, okay, drip shields, housings, okay, related to the equipment. Okay, zone 3 is the wall, floor, drains, okay, air handling unit, the facilities around the food production. And the fourth one is other facilities that is not directly involved with the food production. For example, office area, locker room and maintenance room. So what you can do is you conduct microbiological tests on these surfaces that available in the zone 1, 2, 3 and 4. And the microbial test that usually is conducted is the for the indicator microorganism and also for the pathogens. Indicator microorganism, for example here the total plate count, the coliform, yeast, mold and enterobacteriaceae. Pathogens are for example Listeria, Salmonella, E. coli. Okay, so zone 1 usually we only conduct the test for indicator microorganism. Okay, zone 2, 3 and 4 we also conduct the pathogens pathogen test. Okay, why zone 1 only limited to indicator organism? Because zone 1, if you can see here, these are the surfaces that have direct contact with food. So usually when you test the food and you found pathogens in the food, you already knew that the food contact surface must also be investigated. So that's why in hygiene monitoring, you do not have to conduct the pathogen test. You only conduct the indicator test. So other zone, you have to conduct also the pathogen test. Okay, indicator microorganisms are usually to monitor the overall hygienic conditions. Okay, the pathogens are usually conducted when the likelihood, the severity, and the significant risk is high in the plant, like how you have access in your how you have evaluated in your HACCP. So example, if you run a meat processing plant, okay, you know that salmonella is very relevant to your food processing. Okay, because salmonella love meat. Okay, so you need to test for salmonella in the environment. There are full videos on how to conduct swab test method for the collection of sample from environment. So as you can see in this figure, the swab test is conducted on the surface, and then the swab is brought to the lab to culture the possible microorganism that can be present on the surface. So that is the end of the lecture 12, and please log on to Putra Blast to answer some questions.